Hello and welcome everyone on the call here and some folks here in person. My name is Pat Gallagher. I'm the Advancement Director here at the Trail Conference, and we're excited to have you here in the room and joining us online for a very special presentation from our distinguished guest. Before we begin the presentation, I'd like to point out for those of you on Zoom, you will remain muted during the presentation, and then we will be monitoring the chat throughout. And we'll hopefully have time for Q&A at the end. So we'll return to your questions at that point. So please hold off on asking any questions in the room until that time. And so now to introduce our speaker, Dr. Dorothy M. Preteet is a senior research scientist at NASA Goddard Institute for Space Studies and adjunct professor at Columbia University. She directs the Paleontology Division of the New Core Lab at Lamont Doherty Earth Observatory of Columbia, and in collaboration with the GISS climate modelers and LDEO geochemists, is studying the late Pleistocene and Holocene archives of lakes and wetlands, documenting past vegetational change using pollen and spores, plant and animal macrofossils, loss on ignition, carbon and charcoal in conjunction with accelerator mass spectrometry, radiocarbon dating. Her research provides local and regional records of vegetational and climate history and carbon sequestration. So without further ado, please welcome Dr. Preteet.
Well, thanks for inviting me. Yeah, can everybody hear me? Okay. And can we turn the lights off? You can see it better, or no?
Yes.
Yeah.
Yes.
Okay.
What's that?
Great. Yeah. So yeah, thanks for inviting me. I used to live in Suffolk for 30 years, so I know this area a little bit. And I worked in Alaska for 20 years. I was trained as a botanist, so I love the outdoors, basically. But I had kids and worked at Columbia and wanted to understand the wetlands around here better. So I started working in the Hudson Valley, and those triangles are several of the marshes that we've worked on that I'm going to talk about today. And the picture on the left is Jamaica Bay, which you know is a national park. The red dot there. This is an eroding marsh in Jamaica Bay, yellow bar. You can see how thick the peat is and how it's going into the water. The estimate 70% of that goes back to CO2 and is a greenhouse gas. And this is getting rid of the invasive Phragmites at Piermont, but of course it didn't work. But that's a group of kids working on it. So do I just use the, which one? Oh, just tell you? Okay. Yeah. So here we are. What we do is take sediment cores, and we do it by hand, as you can see. Oh, yeah. We even climb on it. We go down in Piermont Marsh. How many of you know that marsh? You know where it is? I know where it is. That's over 17 meters deep. Okay. Can you imagine? So we had no idea. In fact, an Audubon video says that that was only there when the pier was built. And I remember thinking that can't be right. But anyway, here's Jamaica Bay. We're taking cores. So next slide. It's too bad I have to tell you. I can't just do it, right? Oh, well. Okay, so why do we care? You guys may know wetlands are super important. And a lot of people, well, just in terms of carbon, which is a big deal now, wetlands are 50 times more important than forests, first off. But the reason is they're cold. It's like a refrigerator. They preserve, they're anoxic and they're cold, so they store it. But also they clean our water, they store our water. Think of New York City. It's protecting all those Catskills aquifers. They're nurseries. It's the refrigerator in terms of food for fish, right? And for the food chain in the Hudson. So birds, it's an ecotone. And for us, it's really important when we look at paleoecology. So the history of just plants changing since the last ice age. And you know, the ice age was here. The ice was here in the Pleistocene. They say a mile thick, which is hard to believe. But so we're interested in the whole sequence of what happened after the ice age. What plants came in? Of course, they came in from Jersey, right, from further south. And the birds brought them, and the whole history is pretty neat. And then you can learn about climate change too from the plant history. But wetlands in particular, and wetlands are the places we core because of that storage, right? You can't just go out in the grass and core because it's hard, but you can go back in time. And wetlands are still being lost in this country at a huge rate, despite some of the protections. New York, well, the Supreme Court, you may know, just ruled the Sackett decision. So it's unprotecting lots of wetlands unless they're connected visibly. So, and New York is a real hot spot for sea level rise. That's another reason for looking at these marshes. So the next slide. So the term blue carbon, I don't know if you've heard, is just carbon stored in a marine environment or carbon in a marine. So the plants, the living plants matter too. But there's a lot of carbon in that. And as I mentioned, it's 50 times greater than forests. It's disappearing. And we have a lot in the Hudson that we should protect. So the next slide. So there's some questions we asked. How do they change with climate change? What have humans done to it? What are long-term accretion rates? And how does that change? And how can we save our marshes too? So we get out there and we do it physically. And I like to think of a core when we take it. You can see one here. We have the top meter is a side opening Russian core. It takes a half a cylinder. And then we sample all the way down, depending on what frequency we want to know what happened. So usually a meter core, right, and a meter is 3.3 yards, I think, goes back about 400 years. So, and that is composed, if you think about it, if you've been to a salt marsh, of both mineral material and organic material. And organic just means anything that was living. So plants, animals, insects, whatever. So we divide it that way. And we burn our samples in our methods to see how organic the sample is. If it's super brown, it's probably 80 to 90%. Organic, the rest is sand, silt, or clay. But we also are interested in these others because they tell us about the depositional history that happened in the Hudson through time, and I'll come back to that. This is also important because they have contaminants. The heavy metals are in there. And actually, the heavy metals stick to the organics more than the minerals. But again, thinking of preserving it, we want to understand how the whole thing works, right? The next slide. So if you've been to, how many of you know Jamaica Bay at all? Okay, so you know it's a big bird area too. But here's a map in '88. Actually, I've found maps from, first thing I do is always look at the old maps, the earliest. 1600s, there's a lithograph with little pictures of Native American boats they used that had long ends they could pick up and put in a marsh. It's really cool. But you can see how the natural, all this blue was marsh on both sides. Now it's gone. It's all hardened shoreline, right? Except for the islands in the middle. So we cored, the two cores I took here from Yellow Bar and Jamaica Bay, just to show you then what we can learn, the next slide, from the cores. And a colleague of mine showed the loss that's been happening just in the last 80 years or so as sea level rises and It's frightening, yeah. In Pelham, where we're also working, I think since the 70s, it's lost 40% in Turtle Cove of the marsh. So sea level is rising. The next, so these are the study sites. And the next slide, again, yellow bar. So I want to go over the methods a little bit. So I'm a botanist. So for every flowering plant, there's a type of pollen. And if you're allergic, you probably hate that. I work with the fossils, and so the proteins are gone. But it's really fun because it's learning pollens like, you know, leaves on a tree. They're all different to the genus, not always to the species. And then we look at macrofossils, and that's even more fun because those are seeds. And seeds are unique to the species. They're amazing. If you ever just look at them, they're wonderful. So we look at the macrofossils, we look at foraminifera, which are one-celled animals that are like this, and they tell you where you are in the elevation of the marsh, depending on the species. So I have kids working in the summer counting the forams. They have to count 100 per sample. In the pollen, we have to count 300 grains, which is hard. So it takes a long time. And then we date. You can date one single seed if it's big enough, like one spruce needle you can date. Yeah, and that tells you, you know, is it 400, 800, 1200 years old? And then we burn the samples, which gives us how much organic, how much inorganic, right? Because all the organic burns off and leaves the heavy minerals. And then we also do nitrogen and carbon isotopes, which it would probably, I won't go into the details, but I'll show you, it tells you about pollution, particularly the nitrogen from poop and pee. So, and then finally, the X-ray fluorescence, which is a big fancy machine we put the cores through, give us all the elements. So that gives us lead, copper, zinc, and we can see these profiles through time. The next slide. So here's some examples of pollen. Here's pine, hemlock. You know, hemlock probably around here is dying from the woolly adelgid, yeah. But it means more moisture usually when you find it in the pollen record. Whereas pine, depending on the species, and you can't tell from the pollen, you have to look at the needles, right? White pine has 5, pitch pine has 3. You tell which pine, and then the oaks are there. And what I'm going to get to is that the shifts in the pollen can tell you climate. And particularly from oak to pine is, depending on what the other species are doing too, is usually a drying. And I'm going to show you that. And then these are disturbance species from Europe. So Rumex, and I forget the common name. Some of you guys probably, dock, I think, and also plantago. Plantain, which you see in the cracks in sidewalks. So we can see, next slide, the human impact from those. So just to go into how we tell time, the plants are taking their CO2 from the air, right? So they build their cells, and then we date it with carbon-14 isotopes because they decay, they're radioactive, and you measure how much carbon-14 to 12 is left in them. So we can measure one individual seed. In the old days, they didn't have accelerator mass spectrometry. We send it off to California. It costs $600 to date one seed. So, yeah, Lawrence. You know there? Oh, okay. Yeah, although now they're giving discounts of $300. And if it's a marsh core, actually there's a facility at Yale that will do it for free. So we're trying to do a lot this summer from marshes, not lakes, but marsh. Yeah, so, and then ragweed is a good disturbance indicator because, you know, ragweed's native to North America, but whenever people cut down trees, that's what we see in the lake cores. We see this huge increase in ragweed when the trees decline in the pollen. And then there are other things we can use time-wise, like the nitrogen 15. Often we can see when Europeans come, the nitrogen goes up or population increases. Heavy metal, you know, this happened here in the mid 1800s. It went crazy, all the industry, yeah. But it started earlier. We see it started early 1800s kind of. And then, but we can tie the decline to the Clean Air Act, 1974 or 75. We see lead go down. Yeah, lead. That's the signal. Yeah. Okay, the next slide. So here's a core, right? And here's the inorganic and the organic. So you can see in this particular core from that yellow bar marsh that was eroding that we used to have a lot more sand, silt, and clay, and it's declining. Whereas the organic part is staying pretty much the same. The next slide. And in the pollen, this is how we make a pollen diagram when we finish each level counting. This particular core is 2 meters long. Most of the sites in Jamaica Bay, those marshes are about 2 or 3 meters, sometimes 5 at most. And we do the pollen like every 5 centimeters, right? So what you see here is we didn't have a date right here. We're guessing for that one, but we get a date around 1825 there and a 1543 date here. But you can see the ragweed rising. And then in the 20th century, it falls. And there's a whole thing about reforestation of the Northeast in the 20th century. But what's funny here in this marsh is mostly its grass is increased. And actually, I haven't gone back to that, but Phragmites grass, we can tell from the others by the size, it's much smaller. And I wonder if that's mostly, you know, although the marshes have been so depleted that another hypothesis, which we need to test is maybe that's higher CO2 making more pollen of the grasses, but I don't know. I mean, the allergy people say that's definitely true. There's more pollen now from higher CO2, as well as the season being longer, but we need to do more work on that. And the next one, next slide. Here's the forend. So you can see some of the shapes are a little bit different than what I showed you before. And this is a more low marsh, and then it gets to be a higher marsh. I should talk about the kinds of plants. Do you know in salt marshes you have the very short high marsh grasses, Spartina, patens, disticus, and then near the streams, and when you get closer to the water, you have Spartina alterniflora, which is taller. And that often goes with the remains that we find, although here we did find some alterniflora here as well as, or no, sorry, it was just at the top, which is interesting. But we do that routinely to see if we can take a look at what's happening. You'll also see the loss on ignition curve here. The marshes are about 50% organic at the top, whereas way back here, you can see they were only 10% organic. And the rest was mineral, as I mentioned. The next slide. So now I'm going to show you, we looked at Yellow Bar, Big Egg, and now Joko Marsh. It's the highest elevation still, although this East Meadow is also pretty high. So they're the sort of the most pristine ones left. The next slide. You can see again, it's about 50 to 60% organic at the top, and then goes way down. This goes down to 4 meters here. And mudflat samples have about 5% organic because they're mostly mineral, you know, clays and silts. Okay, and the next slide. Here we've looked at the percent that we burn, so you're getting more organic. But to tell which way it's going, really, you have to look at the independent, see if the organics really increasing or whether it's mostly this decline in inorganic that's pushing that curve. And again, it was the decline mostly. So the next slide. Oh, this was the pollen also. Here's the ragweed rise again, which we know here is in the 1600s when the first people came and cut down trees, and Chenopodium and Plantago increase. We were questioning about drought here. I don't remember what we checked for that. But again, you see the ragweed drop in the 20th century. That's the green arrow. And grasses again increasing there. And some of the trees, I think, were coming back. Yeah, pine comes back and hemlock a little bit, but then hemlock drops again. Okay, the next slide. Okay, now the heavy metals. And this we really need to quantify with a standard. But here's the curve, and around 50 centimeters in all these floors, you see this huge increase in lead and copper. Copper is usually after the lead starts, and then you see the decline towards the top of the core. And the next slide. So this is a paper we published, and I don't have it on here, but it's, I think we published it in 2018 on these two cores and our sort of tie points that we put in for these big changes. So I'll just go over them. Generally, here's the ragweed rise showing the human impact cutting trees. And we know that's around 1650 in Jamaica Bay. The nitrogen, well, the charcoal kind of increases with the Industrial Revolution too. The nitrogen there is presented as percent nitrogen, which are the X's, which increases. That's this curve with the scale going that way. And then the one on top is reversed going the other way, but that's increasing the isotope of nitrogen, 15. And that's a signal also for pollution. So we see in all the marshes a huge amount of pollution with, of course, human population. And then you see the zinc and this 1975 drop when the ban happened. We dated the actual lead in the core, the isotopes of lead, 206 and 207. We sent to someone and they tracked it to Pennsylvania coal to that date of 1839, which is kind of neat. And then this is titanium, and titanium mirrors the mineral content because it comes from uplands. Titanium and potassium are in the soils. So when you see them decline, it's just like the LOI curve that's happening. So basically the story here is that Jamaica Bay, because it's so hardened, has lost its mineral component. So the marshes are sitting there. They have a lot of organic matter. Next slide. But they're falling apart. And they're also falling apart because they have such high nitrogen that the roots, they don't have to put down many roots because they have plenty of nitrogen. So their roots are shallow and weak. And so when sea level rises, it just can mess it all up and make inroads. And that's pretty unique, I think, to Jamaica Bay. It's not, you know, the Hudson, well, it has high nitrogen, but you don't have that big cutoff of mineral sediment. Jamaica Bay used to have 18 different streams coming into that bay. Now there are only 8. And they don't bring much sediment, you can imagine, because you know, it's a hardened shoreline. So the accretion rate there is almost up to 5. And what's interesting is the inorganic is declining. That's the sand, silt, and clay. But the organic matter is actually increasing. And we think maybe the plants look above ground, they're growing better. Maybe they just haven't compressed and decomposed over time much compared to back here. But anyway, it's an interesting. So what they've been doing in Jamaica Bay is adding sediment to the marshes, and that works pretty well. The problem is they've done it with huge dredges from the channels, but then they have to go out and plant by hand. But it is keeping it, keeping it. But they say it's too expensive to do on all of them, so I don't know what'll happen. But anyway, it was interesting just trying to see what the problem, if we could help to understand it. Okay, and the next slide. Now we're gonna, this is sort of a summary. And what I'm worried about with all these is that release of the industrial metals into the water. Because if all these marshes erode, it's in the top 50 centimeters, right? That's the most vulnerable. Then the whole food chain will get it. All the invertebrates, the birds, everything. It's been tied up. So what I go around preaching is if we can keep the marshes there, right? In the Hudson too, then we preserve it from getting in the water, which, we've messed up so much in terms of the world. You guys know that. And so a lot of this, the decision, a lot depends on politics too, unfortunately. So the next slide, I want to jump to Piermont. This is the place that we poured, but we've done a lot of pouring since. And just to see if we got the same thing in the bottom, in the southern part of the marsh, we poured here. And Lamont is up here, I guess a little bit further south. This is Solomon. All right, okay. Next slide here. Looking over it in the winter, this was taken probably 20 or 30 years ago. That's frag, unfortunately. So the whole thing almost now is covered with Phragmites, and I don't think you'll ever get rid of that because it's in every ditch on top of the mountain. And I found papers that show that the seeds of this Phragmites here in the New York area are the most robust. And, because they're hybrids, New York was such a port. So a lot of times Phragmites just spreads from rhizomes, but here it also, the seeds just go crazy. So the next slide. This is when there was some Spartina patens left, and Disticus, if you know, a regular salt marsh is a wonderful place, habitat, right? And this woman was collecting fish, Fondulus. But here's the frag coming in, invading. Next slide. So here's where we took our first core, and a student of mine, Dee Peterson, did her thesis on the top couple of meters of this core. And you can see we get pollen in it from the forest, right? So it's a wetland, but we're getting upland species that fly in there. The next slide. So, and this is just today, the contrast, unfortunately. You can still canoe through it, but it's a monoculture pretty much. Yeah, next slide. So here's the pollen diagram that Dee produced, and this is 2.5 meters. We have a lot of dates from seeds, Here's the change that shocked us. Well, first of all, come over here to charcoal. And what you see is this huge increase, right, in pieces of charcoal, much more than the industrial age or the top. And so we dated it. And what happened is it comes out exactly to the medieval warm time, which, in Europe they liked it because there was grain and they grew all these things. But here, so we go to a big increase in pine, and a decline looks a little more subtle in oak. And then things like hickory, which like warmth and dry increase too. And the hemlock declines. And the way that we know this isn't all one percentage pushing the other is that we throw in an exotic. type of pollen that doesn't grow here, and then we count the ratio of that to individual species, so we know that it's. I don't know if I showed that slide. Maybe the next one might have. Yeah, this is so it's actually the technique is from the mark recapture salamander technique. Do you know that? You go, if you want to know the population of salamanders in a pond, you collect a bunch and mark their tails and throw them back. And then you come back the next week and see how many have the marked tail. And that tells you, that ratio tells you the actual population. So this is a real hemlock decline. This is a real beech decline and an increase in hickory, which tells us it's dry. We just wanted to be sure. And the next slide. So what it tells us is that there was this big drought here, and with things burned. And when we sent the paper for reviews, a reviewer said, well, what about Native Americans? Maybe they just burned the forest or something. But we know it's not. And the reason is the XRF X-ray fluorescence tells us that there's more salt water. So the salt, you know, the Hudson has salt, it's tidal, right? So when you get a drought, the salty water comes way up the Hudson. In fact, Poughkeepsie gets drinking. So we saw that and proved that it was indeed a big drought. But it's interesting because it showed me the vulnerability. We don't think in the East much about drought, right? But in the 60s, if you were, just no, yeah, we had a big drought for only 3 years, I think, and the record droughts went to 4. So it's interesting. I mean, it makes you think. And then this is that same 2-meter core. This is the titanium and potassium and the inorganic. You can see they follow each other. And so whenever the farmers came, they till the soil, right? And with the ragweed rise, you also see this big influx of minerals because they're tearing up the soil, right? And it goes in the river. But way back here during the medieval warm, there's also a lot of sediment. So that seems counterintuitive, right? If you have a drought, why do you have more minerals coming in? But I think what it is that when you do have a drought, everything dies and you get, you know, exposed. And then when it does rain, you get a lot of erosion. At least that's what we think. And the next slide. Yeah, so this is the story about, here's where Piermont sits. Here's the marine elements, calcium, chlorine, bromine, and strontium. And the terrestrial ones are potassium, titanium, rubidium, and zirconium. So when there's high freshwater flux, in the spring, the salt wedge moves out. And then when there's a drought, it moves way up. And it's denser, right? So the fresh is on the top. Okay, the next slide. Tim Kenna is a guy at Lamont who worked on this a lot. He had the first XRF machine and showed this. And actually, we sent in a proposal. We wanted to do the whole core. We've only cored it down to 13 meters, but to see how many droughts we had, right? But we never got funding to do that. So something I'd still love to do. And the next slide, to see the frequency. But the implications are huge, I think. And if you go to the next slide, this is the water supply, right, from in the 60s. When we've been lucky, we haven't, I think in the 80s, there was a tiny drought. But the tree ring record, which you might have heard when Nicole gave a talk here, shows high droughts in the, if you go to the next slide, I think. Yeah, Neil Peterson did a study, and the previous droughts were of greater duration. And so he was arguing from the Hudson Valley that we could be in for a big drought. What's happened since then is that since the 70s, the northeastern US has had a 20% increase in rainfall. So we think we're off the hook, except that with global warming, the models show that that can evaporate. Even 20% more rainfall, you can still have it. So, you know, I think this year we're really lucky with that wet. But it's something to think about, especially because those tree rings really do show some of them were long. And our medieval drought is 1,500 years. It's from 850 AD to 1250. So that's a long time. And then we have lake records further upriver. I just finished a paper on the Catskills, the flora in the Catskills, and there's some big droughts around 5,000 years ago, and the hemlock salt too. So we can, one of the things I want to do is try to tie the marshes to the uplands in terms of what's happening. The next slide. So here's the water supply story again, 90% of the Catskills. And the next slide. Next one, the watershed. Oh, and then we were asked to look at what happens to the fish in the river with drought. And in the next slide, what we concluded, we just looked at research. We see that you get more minerals, right? So they're more inorganics. And the low flow, this is true from Bob, what's his last name, has done a lot of work today on what happens in the drought, or the last 50 years, is that with lower flow, there's more algae because it's calmer and you don't have the flushing out. And so you can get more eutrophication. And but the marshes particularly get less flooded, right? Because it's slow flow. And you can have die-off in marsh grasses too. So it's a big difference for the Hudson. Next slide. Okay, and then here's the Piermont. Down, we've gotten now down to 5 meters, working our way down. And there's a lot of back and forth. It's interesting in the organic part, the percent loss on ignition. And again, this is 10% organic, and that's about 50%. And then the bulk density is just, when you have just peat, organic material, it's pretty light. It's the density is just mass over volume. But when you get into the mineral matter, it gets heavier. So that's just a reflection of that. But we need to finish the whole core. And I'm hoping with 3 dates, we can really do that. Next slide. The top of Piermont, you can see these are all the heavy metals. So lead is green, zinc is yellow, arsenic is red, and copper is blue. And I've got lots of cores up and down in Marsh. Constitution Marsh, how many of you have ever been there? That, you know, the battery cadmium factory was there. And yeah, and we didn't pick, well, I think it was our detector. We didn't pick up that much cadmium. I was glad. But you know, that's the problem. They're all packed with these heavy metals. Next slide. Oh, and here's where we dated seeds all the way down this far. Okay. The next slide. And we made a curve, which here's the top, right? And here's the bottom. And you don't often get the purple is the uncertainty with the radiocarbon age you get. So these all look pretty good. You can draw a curve, but what happens at the core boundary, you can see, I like to show this because sometimes we screw up. When you bring the core up, and then we're using a Livingston piston core, not the side one, things attach sometimes on the side, so you get younger ages. Because this is calendar years over here, and we're going back to 2,000 years ago. But over here, at this depth, we're getting a young age, right? And that just means we picked up something. So we never should take anything from the outside of a core. Next slide. So we measured how much blue carbon these marshes are storing, right? And this is, to get blue carbon, you have to multiply the bulk density by the loss on ignition. So this just shows, you the blue is the bulk density. The sed rate is the solid curve. And the next slide, when I, oh, this is pollen too. That looks better than the other diagram you saw. This is the final diagram. And this is the charcoal to pollen ratio in that core. The next slide, here's the heavy metals in the top. This is now back to only 2.5 meters. Here's the ragweed. Here's the percent inorganic. Here's the nitrogen in this core too. And the next slide, this is the actual carbon flux then. So what's interesting about this, I think, is that the higher rates of carbon storage are back here. Now we have, here's where we are now, only this was like 2010, I think, the top. And we're going back a couple of thousand years. But there was more carbon stored here. But then as you got into the medieval warming, I think what happens, a lot burn off, right? So it's low. In the Little Ice Age, it also is pretty low because I found a diary from Piermont that a man had written, and guess when the first frost was? August. So the season was like August 20th. The season was short, right? To grow much carbon, for the plants to grow, right? And then today we have a higher amount. The blue shadows are the uncertainty. And the next slide. So what we can do, well, yeah, also, if you go back for a second, they were also hay, remember? So they're taking off huge amounts of hay for their animals and houses to chink. And that happened right when the settlers got here. And there are wonderful maps of Piermont that show all the lots of hay for that. And that's a hide, the hay fields, or hay, I don't know how to say it. Okay, the next slide. So this is sort of a summary for Piermont. This hibiscus grows out, or used to grow out in Piermont. I think it's gone. But I think, again, we can calculate the sedimentation rate, how fast it's growing. It's only 2 millimeters a year. And sea level rise is now over like 3.5 or 4 at the bottom. So I think we need to have sediment added again. The Army Corps said no, because it's contaminated. But of course, the marshes are made of contaminants. So I don't see what the problem is. You could also take out dams, which is a win-win, because the eels, fish go up. So those are two things we could do. The rates range from greater than 300 grams of carbon per meter square per year in the earlier time to minimum rates 50 in the medieval warm, which is kind of interesting. And then they slow down again in the Little Ice Age. And then we know, again, that nitrogen increases the above ground, but not the roots, right? So the net is usually less, I think. Okay, the next slide. Oh, this I have had with my students. We burned it down this far. So the blue is water. The orange is the mineral. And the black is organic. So it looks like it didn't vary that much in the organic matter, right? Even though there are different plants growing in here at different times. Because I've looked at some of the spots to get dates. And sometimes it'll be more marine, like there'll be marine seeds of plants, and other times totally fresh algae. So it's flipping around. But you can see at some point here, huge amounts of sediment came down the Hudson. And I have a student who's trying to fingerprint which tributaries, right, they came from, because the Mohawk has a lot of limestone in it. And we can try to pick that up. We don't know what that's made of, but that's where I was saying maybe that drought that we see in the Catskills, 5,000 might match, if we got better dates here, this drought, if this is a drought, I'm not sure, but we're certainly getting lots of sediment coming in. And then, you know, we're that first meter I showed you, or the second one, medieval, I guess it was in the top 2 meters, seems tiny amount of sediment compared to what we used to get down the Hudson. And I don't know why exactly, but it's an interesting question. Okay, the next one. Oh, and then just to show you a lake record, one thing great about the marshes is they grow relatively, you know, 400 years in a meter, whereas all of Mohill back to the Ice Age is only 1.4 meters. So this, you know, a marsh allows you to look at the beach, right? Yeah. But it's interesting too, because here's the percent organic carbon. I did this work with Kirsten Menking, who's working at Vassar, and she's a sedimentologist. So the carbon to nitrogen ratio is greater here, and the delta C13, which is just an isotope of carbon. I think this was a moss in the core down here, yeah, at the base of the core. So next slide. We looked at plant macrofossils again here and saw, I don't know, that's interesting because this is, it does, oh, that's what it was. Yeah, it was a bryophyte. It was called fontanellus. Now I remember it. When we pulled up the core, it looked like red reindeer fur or something. It was so thick. But what we see, what's circled there is a big drought 5,000 years ago. And we know that because we see a lot of, well, hemlock dying, shallow aquatics. There were also sand layers in Mohonk that aren't there now. So it was interesting, all the different seeds that showed up in the skinny little core from Mohonk. Next slide. And then finally, how many of you have been to Iona? Probably more. That core, too, is about, well, Piermont's 17 meters. Iona, we pulled the core about 10 meters. Next slide. And then we went to Tivoli. Tivoli's shallow because Tivoli, well, this part, the peninsula, is old. But if you look at old maps, of course, this is the railroad. So these marshes only formed recent since the railroad. The next slide, I had a student work on this too. And again, you see the inorganics increase in this case in the top, along with the potassium and the lead rise. So there's probably a hiatus in this core from before 1700. The next slide. You still see the trees drop, the weeds rise, and the next slide. Here she's put the pictures. It used to be ferns and some sedges, and now, you know, cattails, most of the cattails that you see around here are invasive. They're favored by high nitrogen and stable water. The diversity has increased, but it's not all a good time. Next slide. So you have 8 times drop of natives and 5 times increase of invasives. And you guys know that's the story a lot around here. The next slide. So if you look at a salinity gradient, of course, you go up and get to quite fresh conditions. And the next slide. Sampisa, who was a student here, did a study of the calcium to zirconium ratio and the calcium to potassium ratio. And then the next slide, the heavy metals. Is this the one? Yeah, well, in Iona, in Piermont, in Sawmill Creek, that's Staten Island, and then in Big Egg. And then the turquoise is zinc, and the black is lead. So you can kind of just see different concentrations, although the scales are very different. Look here. So the highest is clearly Jamaica Bay with that 1,000 ppm concentration. I haven't looked at this carefully in a long time. It's kind of interesting. Yeah, the next slide. I was thinking of this slide though next. If you go up the Hudson, right, from New York City, Staten Island to the Mohawk tributary, what jumps out is near Kingston, where, you know, there's a big industry. That's where the roundout comes out, where they send all the cement and coal, the coal industry. So that's kind of interesting too. The next slide. So the future predictions, again, for sediment accumulation, Scenic Hudson did a study, and they had a low, medium, high prediction for the Hudson Valley for sea level rise. And our long-term rate is, you know, it ranged from a little over 300, I think, down to 50. So we're in trouble. That's my story. We need to protect our marshes. And the next slide. So overall, I've just showed you how we look at what we find in the course, the organic and inorganic, the pollution. Next slide. The arrival of Europeans was really a big deal. And since then, we have all these things like increased nitrogen that are favoring invasives and really changing the environment for the native plants. Apparently, too, do any of you know Robert Nazi at the Botanical Garden? He did a study of plants along the Hudson in the mud flats, and he used older barium sheets to try to go back and find. And there's some plants, I didn't know this, but that were unique to New York, and I mean unique in the world. And some of those are gone, and he thinks it's all the nitrogen kind of, because that's why frag gets such an advantage. This is just a core we took from Staten Island. You can really see the difference between the organic, and then it gets siltier. The next slide. And so these are just the human impacts I think we went over. And the last slide, I think. Oh yeah, we're facing warmer temperatures, right? We're facing greater storm frequency. So we know the Catskills have had big erosion. We know sea levels rising. We know if you dredge, you do get remobilization of pollutants. But I think we should spend money on nitrogen removal, because that's a win-win, right? For plants, for the water, and we could add sediment to the marshes, and we could think about, and Scenic Hudson has done this, and DEC too, I think, locales for marshes to migrate.
Migration.
But the Hudson Valley is narrow.
Yes.
Yeah. And the railroads too on the east side. So that's a problem. And the next slide. Oh, I was just gonna, some pictures of the restoration are nice, I think. And then the next slide is just dams. This is amazing. This is all the dams. There are over 700 in the Hudson waters. A lot of them are old on little streams, you know, but people don't want them taken out. They like their little. So I think that is a problem. And then how many of you know the Croaks there? It's amazing thing to see. Of course, it's leaky. It's scary to go over there. These big rocks and the water's coming through.
Yeah.
But that's my last slide. Yeah, thank you for listening. Question.
Yeah, we have a question online.
Sure.
You talk about hemlock pollen counts. Were you able to see increase in hemlock palms around the 1800s when the tanning industry became common.
Yeah, we do. Yeah, good point. We'd see a big one. And I hope to do more coring up there to see if I see more. Yes.
So how would you nitrogen removal?
How?
Yeah.
They do the tertiary treatment plant. Somehow they take it out, and I don't know exactly how. It's a good question. I want to go through one. You know, you can go through sewage treatment plants and they'll teach you, but it's expensive. I think that's the problem. And I don't know how they do it. They probably, yeah, I don't know. That's something I'll look at. Yeah. Good question. And some communities do, you know, Long Island Sound, we're now coring some marshes. They had that huge dieback. Do you remember when they had in the western part of the sound, the lobsters were dying and
Yes.
the dead zone? So they got all the communities together and they cut their nitrogen by a huge by doing tertiary treatment. Yeah, and I think they also worked a lot on septic, you know, putting in treatment instead of just homes that had all septic. And Jamaica Bay has gone down too, but it's just still so much.
Was that primarily because of like all the PA rules that came in?
So yeah, I think they forced it. You know, Jamaica Bay has like 6 landfills around something.
Yeah.
It's unbelievable. And some of the landfills are and the landfills are emitting methane too. To do, yeah.
Several years ago, I was at a talk at Blackrock.
Mm-hmm.
They were talking about doing core samples in the Sky Lakes. They said that they had evidence of a meteor strike in the Hudson Valley. I was wondering if you saw any other influences of the environmentals.
Yeah, I'm skeptical of that. I've worked in Blackrock too, and the Skylight, you know, Mohawk and Minnewaska report too, and we don't see evidence of that. But maybe I found a paper the other day. We count when we look at macrofossils, right, in the cores, and we find these round. You know what sclerotia are that fungi make? The perfectly round black things. And we found, I was working with a student, we're working on the bottom of the Greenland ice floe now. We're finding all kinds of cool things. They drilled the Greenland ice floes, Camp Century, and underneath is the soil that has sclerotia and all kinds of plants, mosses.
So you say now on salt marshes, 1 meter is 400.
Yeah.
And then you set up at Mohawk, it was like 1 meter was
15,000.
15,000. So what is it in Greenland?
In Greenland, we can't, radiocarbon only goes back to 50,000. So they've dated, actually, it's about, I don't remember, I think it's about 1 meter of sediment. I haven't been able to get pollen on it yet, but we found, and the mineral people can try to date it with something. There are 2 ways. One is beryllium-10, which accumulates in quartz from the outer, you know, it's cosmic. And they say the top part of that core is over 400,000 years, and the bottom is not greater than 2 million. So we're finding all these neat plants, like juniper had a stump for the longest time, these tiny little pointed things, which were too thick for mosses. And I'm used to finding spruce and fir and pine, and I'm not used to working in junipers are pretty dry. Often they're not in lake sediment so much or marshes. Finally, I was walking by a plant and said, oh, maybe it's juniper. And sure enough, it matches. That's what's fun. You match it today, right? So yeah, those ice cores are old. They're pretty amazing what you find in them. We found isoedes. Do you know what that is? It's a little aquatic plant that grows at the side of the road. But all of them are pretty much Arctic, except I didn't realize juniper grows way up in Greenland, but it does.
Maybe just didn't, I think it does now.
It goes pretty high. I think it goes to 73 degrees. That's what the student said. I haven't looked, but you know, it's pretty hardy. But you think about birds bringing things and things getting around pretty fast.
You ever find it exciting in parks?
Yeah, we do. And I know one beetle person who's in North Dakota. And are any of you beetle people? What I thought were beetle parts, which were, I wish I had, oh, I could find it. Really cool, like a head, skeleton, carapace, I guess it is, and then wings, you could tell. But he told me that what I was thinking were beetles were all crane fly, which, you know, they're bigger. Well, some beetles. But yeah, we find all kinds of neat things. Yeah.
So we have another question. Are inland marshy areas similar to wetlands storage?
Yes, a lot of them are. Yeah. So we're trying to understand that now. There are not that many people who've been adding up the carbon. You know, and seeing what the accumulation rate is over time. But a lot of the inland ones are, yeah. And they're not protected. That's the problem with the new septic decisions. But I'm sorry, I got sidetracked. I was thinking those round balls that are fungal resting stages for the fungus. When things get tough, when it's not moist, you can't make mushrooms. They make these balls. And my student found a whole book on them that said that people thought they were from outer space for the longest time. And there were papers and papers written on the Younger Dryas comet explosion because they found these black sclerotia that we always thought. So yeah, people thought they were from outer space. So I'm skeptical of outer space. I don't know. I mean, you know. But we have some cores from black hole. There's some neat. In fact, we're writing a paper comparing the lake record to a bog record right next to it. So it's interesting to see what gets preserved and how the pollen differs. Because a lake collects more like pine, you know, you see it on the farm. Whereas the bog would only get what's in one little spot. So that's another thing we're working. But it's the main reasons to get outside. So we get outside and see what's out there. Yeah.
You do any pollen concentration when you're doing your studies, or
Modern concentration, or the pollen influx is what I showed you where we throw in the exotic. So that gives concentration of each type.
Yeah, but I mean, when you're taking it out of the sample, concentrated somehow.
Oh yeah, it takes 2 full days to do 12 samples. So what you do is you take a cc of sediment, and then you put in hydrofluoric acid to get rid of sand, hydrochloric for carbonates. Then you use something else, a mixture of chemicals to get rid of the organics. So that at the end, and you're centrifuging and washing all this stuff, at the end of the day, you have a concentrated pollen. And that's what we put a drop on the slide and count by 100. So it's a real pain in the nuts. We screen it with screens. The sizes are 7 microns, the lowest pollen, and 150 microns. So it's that size.
In your report, burning the organics, I mean, can you burn everything off or does it turn into carbon or do you?
It turns into CO2, I guess. Yeah,
Or do you see if we don't get any like charcoal left over or anything?
we never have. We burn it at 500, so I guess that volatilizes all of it.
Yeah, we have Leola asks, how can we get our marshy and forest area carbon sequestration management? Is there students who would like to do this on our land mass or what land is this?
Yeah, it's easy if you just have a core. Well, you can measure how much carbon without dating it, right? You just have to core it somehow. So you have to have a core. And then it's easy to measure. You just take one little crucible, you know, those ceramic, and you put a cc in there, and then you dry it and weigh it again. That tells you how much water. And then you put it in the high oven for at 500 degrees for an hour and weigh it again and just subtract, and that's your organic matter. And then you just add it up based on the how much is in a core, right? And extrapolate out to the whole marsh, and that's your number. So that, I mean, there are a lot of people doing it now to compare, well, because of the whole carbon credits and the whole economic thing now, carbon trading. And some towns wanted me to do it, Constitution, so they could subtract it from their town off, you know, as an offset. And I said, no, you didn't start this marsh.
No.
You can't do that. But that's how people are trying to use it.
Greenwashing.
Yeah.
That's what.
So yes. And they're doing that with forests too.
Right.
But of course, a forest won't last. You know, trees fall down and oxidize and insects eat them up. Marshes are much better at storing.
Yeah.
A question for you guys. Are you connected to the striped invasive?
Yes, yes.
You are.
Yep,
Is it you guys?
Yes.
Is it? What's your range? North, south. Really?
Lower Hudson Valley.
Lower Hudson Valley.
Oh, you can't be in the medium Hudson Valley? It just, what's the boundary?
I think the boundaries. Like the northern. Yeah, I remember exactly. Yeah.
But is there somebody who does it in the Catskills?
Yeah, so New York State has it divided into sections, so there's like a Finger Lakes, strike forest, there's Catskill strike forest, there's a Taconic strike forest.
So, yeah, a while ago, I was working in Harriman Park and we were trying to get rid of barberry. And they, you know, so we cut it and then called in the invasive strike force to paint the bad stuff.
Oh.
Was that Roundup that they painted?
Yeah, they used to say it for barberry.
But it's not good to breathe, right?
No.
Because you wear a mask.
My understanding is that they did.
Because there's a lot of barberry. I just moved from Suffolk last year to Potterkill, which is near Kingston. And the rail trails, there are a lot.
Rail trails.
And the barberry is just.
Luxury.
Pardon?
Luxury.
Yes. And I know a way to get it if it's small. Do you know this trick? You put on double gloves and pull the branches towards you and all the leaves come off. You know, because it's the way the thorns are facing. So you do that. Yeah, a guy at the botanical garden told me. And the small ones, you can just denude it and it'll try to put some back, but you can get those. And then the whole thing dies. You don't have to use chemicals. But the problem is there's so many big ones. So
Have you gone to Doodle Town?
I have gone.
All right,
Yeah.
that's Barbary Central.
It's natural, there's a.
No, central.
Oh, central, yeah. Yeah.
Central. No, after all the. Houses and cabins were taken down. Or they're
Harriman's so.
Yeah.
Are they doing, I mean, we organized a group and then called the strike. But I don't know, but that was just in one camp. The ACA. Surveyed it, but yeah, it just seems like that's the only way. Because it's taking over. There's one trail, the O&W trail I've walked on, that has wonderful ravines, a trillion. But the barberry's coming and it'll wipe it out, I think, if we don't get it.
I know it's almost lost the priority.
Yeah.
Like it's triage now.
Yeah.
No.
See, that's the problem. It's triage.
But I called the town, I'm in the town of Marbletown. They would not give me the name of the people who do the trail to contact. They said we can't give their emails, which we'll get back to you. But that was back in April. So I'll have to go over again. Because it grows so fast, right? Have you seen kudzu around here?
It's come. Yeah, we can tell it's coming out.
Also, since city, I know the Bronx has a lot of issues with, but it's Japanese not.
Yeah, I don't know.
Yeah, because even a piece as small as a thumb can reproduce a whole plant because it's a rhizome base.
Do theory not leave them?
I wonder.
It's.
I heard about not leaving sort of all everything.
It might be an insect.
There's a few bacterial species that can be injected into the stomach.
Yeah, I thought they had some way of controlling that better or not.
But one strategy that at least the Bronx River Cooperative was using was covering it in plastic bags. And then basically once it's starved and really wants water uptake, then they directly inject glyphosate into the stem. Wow. And then it just sucks it in the whole. You're just trying to get any water it can.
They go laterally.
Yeah, so basically it spreads really easily when it's on a riverbank and it rains. You have a hard rain that causes soil erosion. Because then the pieces will break off because it's segmented in its rhizome. And so then it will flow it on the river instead.
Just so much here. And there's more coming. That's the thing.
A lot of people. We can attack it.
Yeah, it depends. But you'd have to, I guess that's what they do at strike forces, they pick priority area, how they get picked. And which species you want to do. I mean, like I have a president of a little nature sanctuary in Tenafly, Alpine. And we don't have any barberry because our priority, but this was a decade ago, was we look, as soon as we see it, we get rid of it. All right. So and that was a priority to get rid of. There are other things, but at least that was no. I mean, there are things, still grass coming in. I mean, forget about that. In 5 years, it covered all of North Jersey forests. Took 5 years. Boom.
I think that shows one of the important things is really the regulation of landscaping, because that's where a lot of these plants escape from. It's from planted installations. In New Jersey.
In some.
I don't know if it's legal to sell anymore in New York State. So I know New York State has cracked down on several, like Japanese stillgrass is illegal to use as a landscaping area in the state of New York.
It's all here now. We have less ferns. The fern 20 years ago, with the ferns everywhere, beautiful. And now still grass. Still grass by the ground. With it, it's interaction of things, you know, one thing was, you know, well, where we are in Bird County, you know, the deer is like the keystone species, and it basically took out the other story. The heavy rains, we had heavy rain, but it's worse because the understory, and then all the deer like a lot of native plants, a lot of invasives that don't. So, and of course, a lot of the invasives coming in are just hardier. They can, they're better competitors, quote unquote. But it means, I don't know, you know, somebody's gonna know what's now, when do the non-natives become natives? I have no, I'm asking questions now I never thought about asking.
I think the bottom line is a monoculture.
Yes, I get that. Or, yes, that's true.
Because they don't.
It's like frag bodies, your frag.
Is that now they're using for hemlocks for the woolly and velvet. There's something about that eats the velvet, but I don't know how widespread.
I think people are trying to see lots of things and then what will work without causing even more problems.
But you saved your place from Barfield.
Yes.
I mean, because that's right, you can get the little.
That's what we do. Yes. Then we have a whole list of, and especially if something comes in new, you know, there were some of these things that came in with the past couple of years, and so we'll say, okay, try to make sure, okay, that one will, whenever we see it, we'll remove it.
There's a book I read, just now I'm forgetting the author, David somebody, but on oaks as a keystone species. Have you read this book? That one oak tree has 150 insects.
That's right.
And so when you think of feeding a bird, feeding their babies, I never thought about it. They don't feed their babies seeds from our feeders. They have to have insects. So they're saying, you know, people just, instead of a long planted one oak tree, it does so much year round in terms of.
Oh, I agree. The thing of what plant oak though in our area, you've got to protect them because the deer just take them.
Especially light.
Yes. Yeah, but oaks and that's one thing. Yes, they do. Yes, they do. That's another issue with the oaks. Well, they're also talking about there's reasons why oaks are declining in that area, but certainly this overgravity pattern has not helped. Yes. Yeah. What are coyotes?
The deer population.
They're out here.
Now there's a lot of bears, the bears evolving.
Yeah,
The bears and the coyotes, foxes.
yeah. I don't mind that. Deer, I think they've done studies. If you think of per acre, we're at 6 times.
I noticed, yeah, I used to look.
It's huge.
So, yeah, much fewer here. For
Really? They are everywhere, I think.
yeah, and then people hit them.
Well, that's leading cause of death, yes.
For deer?
No, for humans.
No, for deer in New Jersey.
Oh. Yeah.
Deer?
Yeah, here, yeah, that's great. Yeah.
It's, you know, something in the
No, they don't understand. They just don't understand like predator are dangerous. They don't. They've never learned. Maybe it's, well, deers are.
You'll see like the
It's very true.
Maybe this is a salt marsh. We love going around.
All right.
Yeah.
It's everyone online. Thank you so much. We'll be recording this.